The process of oxidative phosphorylation is where we finally get to see where all those electrons are taken to by the electron carriers NAD and FAD. Oxidative phosphorylation is broken up into two components, the electron transport chain and chemiosmosis. The oxidative part of this term refers to the electron transport chain where oxygen is required, and phosphorylation describes the role of chemiosmosis. The electron transport chain occurs in the inner membrane of the mitochondria. It helps to produce the 26 to 28 ATP by oxidative phosphorylation via chemiosmosis, but the electron transport chain itself does not produce any ATP. Chemiosmosis is where all the ATP is produced. In chemiosmosis, this refers to the term chemical push. The chemical that's pushed across the inner membrane is the hydrogen ions or protons that are dropped off by the NADs and FADs, and those ions are pumped across the inner membrane of the mitochondria into the intermembrane space where they accumulate in high concentration. All of those protons will flow back down their gradient or diffuse across the inner membrane through ATP synthase, and as ATP synthase spins, it will open up catalytic sites to be able to combine inorganic phosphates with ADP to make ATP. Thus, in cellular respiration, the majority of the ATP produced occurs in oxidative phosphorylation. The electron transport chain, as you can see on the right figure, contains a variety of molecules that are embedded in the inner membrane of the mitochondria. And the mitochondria inner membrane has a lot of those folds or cristae to be able to accommodate a lot of these molecules. These molecules are tightly bound protein and non-protein components. You can see that they're purple in the diagram to represent they are proteins. And as electrons are dropped off by NADs and FADs, you can see in the picture where they're located with the orange boxes for NADs and FADs. You can see those electrons are dropped off at these molecules. And as these molecules gain electrons, they are reduced, and then they will pass these electrons on to the next molecule next to it, and they become oxidized. So all of these, so basically the electrons are passed from molecule to molecule in oxidation reduction series. And so you can see that the amount of free energy starts to drop because there's energy released as electrons are passed from molecule to molecule, and that eases the fall of those electrons. So small amounts of energy are released at once, at small bits, and not all at once, so you don't combust in a big ball of gas and fire. And so that electrons go from glucose or your food in small steps all the way out to the electron transport chain, where the final electron acceptor is oxygen. And when oxygen picks up those electrons, it also picks up protons, those hydrogen ions, and forms water. So the majority of water produced through cellular respiration occurs right here because oxygen accepts those electrons, and the main byproduct of cellular respiration, that water, is produced right here. In this diagram, you can see sort of a side view of the inner membrane of the mitochondria. You can see that those purple blobs are the molecules that are embedded in there that make up the electron transport chain. And I want you to follow the electrons with the yellow arrows. You can see that NADs drop off at the complex 1, and FADs drop off at complex 2. Does that make a difference? And the answer is yes. So as those electrons are passed from molecule to molecule, energy is being released. So there's actually more energy being released when electrons are dropped off by the NADs because they're dropped off earlier in this electron transport chain. The energy that is released by the redox reactions of these electrons being transferred drives hydrogen ions, or these protons, being pumped across the membrane from the matrix into the intermembrane space. And I'm going to say pumped because They are pumping from a low concentration in the matrix to a high concentration in the intermembrane space. And so you can see, as we've done in our drawings, all those protons are collecting there in the intermembrane space. Now, fads also drop off electrons and protons, but it drops off later on in this chain, so less energy overall is released by the electrons that are dropped off there, so not as many protons can be pumped into that space from fads as you do nads. Ultimately, as you follow the pathway of electrons at the end of the yellow brick road or the end of the yellow arrows, you can see that the final electron acceptor is oxygen at the very end. Oxygen is highly electronegative. It loves those electrons. It's going to take those electrons and protons, and when they combine together, it will form water. And so water is the waste product. So you can see here, without oxygen, there's no final electron acceptor, and all those nads and fads that are loaded up with electrons have no place to offload their passengers. So cellular respiration, to fully get the impact of all the ATP you can out of it, you need oxygen to be present and available. So this is aerobic cellular respiration. The final part of oxidative phosphorylation is chemiosmosis. This is an energy coupling mechanism where the energy released in the electron transport chain drives the pumping of protons into that intermembrane space. And so that pumping across is called chemiosmosis. It creates a proton gradient across the membrane, and that storage of protons there creates a gradient that is stored chemical potential energy, and it can drive cellular work. So because all of those protons collected there in that space, that creates a proton motive force. So these protons, these hydrogen ions, can actually move, and as they flow and move and diffuse down their concentration gradient, it can be used to perform work. And so that's a chemical storage of energy, and the work that it can do transforms into mechanical work. So you can see here in this diagram on the right, that is ATP synthase, the enzyme that catalyzes the reaction to make ATP. It actually has a physical rotor-like component, so as protons are flowing down their gradient or diffusing back down across the membrane from the space into the matrix, the only place it can go through is through ATP synthase. And as those protons flow through ATP synthase, it starts to spin. There's a mechanical movement there. And as it spins, it opens up catalytic sites that allows phosphates to be able to be joined with ADP to make ATP. So you can see that in the diagram, lots of ATP produced. And so we're going to use the energy from the proton gradient, and as it flows back across the membrane from the intermembrane space into the mitochondrial matrix, we can use that to drive ATP synthase, thus the proton motive force. Finally, let's put everything together. Chemiosmosis couples the electron transport chain to ATP synthesis. So now you can see the full picture. On the left, you see the electron transport chain with all the molecules embedded in the inner membrane. You can see the electrons being dropped off by nads and fads in different locations. And you can see the pathway of electrons in the yellow arrows. And as the electrons are dropped off, so are the protons. And the energy that's released from all those redox reactions of the electrons being passed from molecule to molecule, that energy release drives the pumping of protons across complexes 1, 3, and 4. And so there's a high accumulation of protons in that intermembrane space. The only place where they can flow and diffuse down, and you can think of it as relieving pressure as well, is the only way they can flow is through ATP synthase, which is also embedded in the inner membrane of the mitochondria. And as the protons flow through, ATP synthase spins and moves, and catalytic sites open up, ATP is produced. So you see here, electron transport chain requires oxygen, chemiosmosis does the job of phosphorylation, so all of this together is oxidative phosphorylation. finally, the last part of our lesson on oxidative phosphorylation is why textbooks publish a variable number of ATP being produced, that during oxidative phosphorylation, 26 to 28 ATP could be produced. So let's go back to this diagram and take a look at all the math involved. Where are all our ATP coming from? So in glycolysis, our net gain of ATP is 2, and that's gained by substrate level phosphorylation. Pyruvate oxidation doesn't have any ATP produced. Citric acid cycle, we can produce 2 ATP by substrate level phosphorylation by the complete breakdown of glucose. So all glycolysis and citric acid cycle, not a ton of ATP. Now, oxidative phosphorylation in the purple on the very right side shows about 26 to 28 ATP, and that's a variable number. Now, why? Well, I want you to take a look at the top here. And so you can see that all of these electron carriers that are involved in moving electrons over to the electron transport chain are your NADs and FADs. And in pyruvate oxidation and citric acid cycle, those NADs and FADs are made in the matrix, and they just get, moved over by diffusion, or they're bumping around and go over to the electron transport chain. That's fine. They drop off their electrons, get their products at the very end. We get ATP. However, glycolysis does offload electrons onto NADH, and that NADH cannot cross the 2 layers of the membrane of the mitochondria. So you can see that the NADH, they get close to the membranes of the mitochondria, and then those electrons have to be transported across. And on the other side, inside the mitochondria, the shuttle carriers or the electron carriers or electron shuttles that are waiting could either be NADH or FADH2. So if those electrons that kind of get ferried across into the matrix of the mitochondria, if they get picked up by NADH or NADs, then we know that we can pump more protons into that space and get more ATP out. So if that's the case, and those little electrons are moved onto the NADs, then we can get up to about 28 ATP in oxidative phosphorylation. However, if those 2 little electrons are shuttled onto the FADs, and the FADs go to the ETC within the matrix, then you get a little bit less ATP out of that. So in total, about 26 ATP. So overall, if you count up 2 ATP in glycolysis, 2 in citric acid cycle, and 26 to 28 ATP in oxidative phosphorylation, in sum total, cellular respiration can produce a total of about 30 to 32 ATP. So this diagram shows you that ATP yield varies depending on which electron shuttles are moving those electrons around.